Hello everyone, myself Janhavi Gedam, student of second year Pharm. My topic for today's presentation is Bordetella pertusis. So, the content of the topic is first we will see the introduction, then hierarchical classification, history, morphology of bacteria, etiology, mode of transmission. Then we will see how it affects the human physiology, signs and symptoms, diagnosis, treatment and the prevention. So let's start with the introduction. Here we can see in the picture that a child is having a cough and trouble breathing, and this disease is called as a pertusis. Pertusis is a severe respiratory infection that affects both the upper as well as lower respiratory tract of the humans, and it is particularly severe in the infants of developing countries. Bordetella pertussis is a small gram-negative Coccobacillus bacterium which is the causative agent of the pertussis, also known as whooping cough. The species name pertussis is taken from two Latin words, per meaning very and tussis means cough, which represents a severe cough caused by a bacterium Bordetella pertussis. The incubation period. Now what is incubation period? It is a time gap between the entry of pathogen and development of symptoms. It is 10 to 16 days for this disease. Due to a persistent coughing, the air inside the lungs has reduced considerably, and whenever child breathes, the air comes in the lungs very rapidly, which creates a whooping noise, which gives pertussis its other name, that is the whooping cough. Let's see the hierarchical classification. Bordetella pertussis belongs to domain Bacteria, phylum Proteobacteria, class Beta Proteobacteria, order Burkholderoides, family Alkaligenaceae, genus Bordetella, and the species Bordetella pertussis. And the binomial name for these bacteria is Bordetella pertussis. Now we will see the history of this disease. The disease pertussis was first described by the French physician Guillaume de Bailleul after the first pertussis epidemic of 1578. Epidemic means the large number of people suffering from the same disease at the same time. The causative agent of the pertussis was identified and isolated in a pure culture by Jules Bordet and Octave Janhavi in 1906. Now, where this bacteria is found? So, let's see the habitat of the pertussis. The primary habitat of Bordetella pertussis is a ciliary epithelium present in the respiratory tract of the human beings. The occurrence, however, has been found in respiratory tract of the human. Bordetella pertussis has no known reservoir other than the humans, and bacteria are assumed to transmit directly from one infected individual to another. The bacteria does, however, survive in air-forming aerosols or droplets. Aerosols is a suspension of fine solid particles or liquid droplets in a gas or air, example fog, dust, mist. Let's see the morphology of bacteria. Bordetella pertussis is a small, rod-shaped, coccoidal or ovoid bacterium ranging in size between 0.2 to 0.5 micrometer in diameter and 0.5 to 2 micrometer in length. It is encapsulated and does not produce spores. It is a strict aerobic or obligate aerobic bacteria, which means these organisms require oxygen to grow, and it cannot be grow in absence of oxygen. As the Bordetella species are gram-negative, they contain an outer membrane and inner membrane, in between which is a cell wall. The cell wall is composed of a single layer of peptidoglycan consisting of glucose derivatives, N-acetylglucosamine and N-acetyluramic acid with pentapeptide linkage. The outer membrane is composed of lipopolysaccharides. The lipopolysaccharides of Bordetella pertussis acts as a endotoxins that are the toxin to the host and thus act as a virulent factor. It is arranged in singly or in groups. Bordetella pertussis is non-motile with no flagella, and the bacteria are nutritionally fastidious and are usually cultivated on rich media supplemented with blood. Let's see the etiology. Now, what is etiology? It means the study of causes of disease. Pertussis, a respiratory illness commonly known as a whooping cough, is very contagious disease caused by a type of bacteria called as a Bordetella pertussis. These bacteria attaches to tiny hair-like extensions called as cilia, which lines the part of upper respiratory system. The bacteria release toxins or poisons which damage the cilia and causes airways to swell. Let's discuss the pathophysiology of disease, that is the mode of transmission. This disease can be transmitted from person to person by droplet infections and direct contact. People with pertussis usually spread the disease to another person by coughing or sneezing or when spending a lot of time near one another where they share breathing space. Many babies who get pertussis are infected by older siblings, parents, or caregivers who might not even know that they have the disease. Now let's see how this bacteria affects human physiology. So this picture shows that how infection may develop in a person or in a child. In first step, child is inhaling a bacteria, Bordetella pertussis. In second step, after inhaling the bacteria, cilia sweeps away foreign object. Then bacteria reproduce and migrate towards the lungs. Then bacteria releases toxins and kills the cell. In another step, toxin released by lungs spread throughout the body, and in last step, pneumonia develops when the lungs get infected. Let's see the signs and symptoms. Early symptoms are runny nose, mild occasional cough, apnea in babies. Apnea means a pause in breathing or difficulty in breathing. Then later stage symptoms are the paroxysms, exhaustion means feeling tired. Disease progression. The catarrhal phase of the disease is non-specific mild clinical syndrome which lasts from 1 to 2 weeks. Symptoms of pertussis at this phase are runny nose, low-grade fever, mild occasional cough, and the respiratory secretions are the most infectious during the catarrhal stage. Then the paroxysmal phase, which usually lasts 2 to 6 weeks, and the paroxysms are the repetitive series of 5 to 10 or more forceful coughs during a single expiration followed by a sudden inspiratory efforts leading to the characteristic whoop. The convalescent stage, which lasts 1 to 12 weeks, is characterized by decreasing frequency and severity of the coughing episode, whooping and vomiting. Let's see the diagnosis. Diagnosis of pertussis is difficult as the symptoms of disease are similar to the other mild infections like the common cold. The most important and useful marker that can be used as the indicators of the disease are a mild increase in leukocyte count and marked leukocytosis. Diagnosis of pertussis can be obtained either through a traditional method of culture and a serological testing or by the use of genomic analysis to identify the organism accurately. The sample collections. Sample commonly used for diagnosis of pertussis are nasopharyngeal aspirate or nasopharyngeal swabs. Then a serological testing. Serological testings can determine whether a person has been exposed to particular microorganisms. A blood test to detect the presence of antibodies against a microorganism. We know microbodies are the proteins our immune system makes to fight foreign substances. Serological diagnosis of Bordetella pertussis is based on the identification and variation of immunoglobulin G or immunoglobulin A against different virulent factors of Bordetella pertussis during the acute and convalescent phase of the disease. Then PCR. PCR test means polymerase chain reaction, which is used to detect genetic material from the specific microorganism. Diagnosis of pertussis by PCR, that is the polymerase chain reaction assay, is an established method for the detection and identification of causative agent of pertussis. Now we will see the treatments. The treatment of pertussis can be obtained either through the use of antibiotic therapy or by vaccine immunization. The early treatment of disease with recommended antimicrobials is useful in the prevention of harmful complications and the reduction of duration of disease. Some antibiotics are used to treat pertussis are erythromycin, azithromycin, etc. The first choice for the treatment of pertussis has been the administration of oral erythromycin. The dose of antibiotics for the children is 40 to 50 mg per kg given to every 6 hours for 14 days, and the administration of erythromycin reduces the symptoms during the early course of illness. Then azithromycin is a newer antibiotic for the treatment which is used 10 mg per kg on day 1 and 5 mg per kg on day 2 to 5 as a single dose. Now, preventions. The most important and efficient method of prevention of pertussis is by the administration of vaccines that are either whole cell vaccines or acellular vaccines. Vaccination is a simple, safe, and effective way of protecting ourselves against harmful disease before we come in contact with them by using our body's natural defenses to build resistance to specific infections and makes our immune system stronger. Whole cell vaccine DTWP and the acellular vaccines are DTAP can immunize the infants. Also, this vaccine is used for the treatment of diphtheria and tetanus. Besides, other methods like the use of masks, maintaining appropriate distance with infected individual is important. Thank you.